You must have probably been wondering what nutrition actually means. So nutrition is the process whereby you take in food and that food produces energy for you. It's as simple as that. And it undergoes some certain processes like the digestion, where it will now pass through your anus, and all those are your physics, right? So that process where you take in food and that food was able to produce energy for you, that's what they call nutrition. And nutrition is of two types, which is the autotrophic and the heterotrophic nutrition. So the autotrophic nutrition is the type of nutrition whereby plants manufacture their own food, and heterotrophic is whereby you are not able to manufacture your own food but depend on other sources of food. So let's look at the autotrophic nutrition. Autotrophic nutrition is the process, like I said, whereby plants manufacture their own food, and they are of two types, which is the photosynthesis and the chemosynthesis. So this photosynthesis is the process whereby green plants manufacture their own food in the presence of sunlight, and this photosynthesis take place in the chloroplast of the leaf. The chloroplast is what contains the green pigment known as the chlorophyll, and it make use of inorganic compound like your CO2 and your water, and in the presence of sunlight, like I said. And the end product of photosynthesis is glucose, and you can also call you glucose your starch. I want to give you one question alongside my teaching, and I'm going to tell you the answer. So plants store sugar in the form of starch, yes, and animals store sugar in the form of the glycogen, yes, that's correct. Now I want to give you how you can be able to detect the presence of starch in a leaf. Yes, how you can be able to detect the presence of starch in a leaf. First, you will boil the leaf. When you boil the leaf, it makes that leaf die. You are actually killing the leaf. So when you boil the leaf, you kill the leaf and then remove it from the boiling water. Then place that leaf in hot charcoal to decolorize the leaf. Decolorize the leaf in hot alcohol. Then after you decolorize that leaf in hot alcohol, you remove the leaf from that hot alcohol. So the third step to do is to place that leaf in boiling water. When you place that leaf in boiling water, you remove it. So that the leaf that you place in the hot alcohol is to actually remove the chlorophyll in that leaf. So when you place it in hot water, remove it. After you remove it, place it in iodine solution. So if the leaf turn blue-black, you know that starch is present. But when the leaf turn light brown, know that starch is absent. So that's just how to test for the presence of starch in a leaf. Now, photosynthesis occur in two stages. Yes. And they are the light stage and the dark stage. The light stage is also known as the photochemical stage. And in this stage, they have many processes. Number one process is the activation of chlorophyll. That's the number one stage in the light stage of photosynthesis, the activation of chlorophyll. The number two is the splitting of water. The splitting of water. You know water formula is H2O, yes. And when you split it, it will give you H plus and OH minus. So when you split that water, that's the second stage of the photosynthesis in the light stage. Then the third part is when oxygen is being given off as the byproducts. Yeah. So oxygen will be given off as the byproduct in the third stage. Then the fourth stage, that H plus that was split in that water will now be transferred by the NADP coenzymes. The NADP stands for the nicotinamide adenine dinucleotide phosphate. That's the meaning of NADP. So the last stage is when the ATP is formed. Okay, so that's just it though. And let's move on to the dark stage. The dark stage is also known as the biochemical stage or the Calvin cycle. The dark stage is also called the dark reaction stage because it occurs in the absence of sunlight. Yes. So let me tell you the processes in this dark stage. The first one is CO2 being reduced. Yes, and the CO2 being reduced is then combined with the H plus ion from that water molecules, and that H plus ion is produced by the NADP. The NADP, what did I say NADP stand for? Nicotinamide adenine nucleotide phosphate. Yes. So that's it, phosphate, sorry. And this, after it's combined with it will now produce sugar. The energy needed in this dark stage from the production of sugar is gotten from the ATP in the light stage reaction. Yeah. So what I'm going to tell you now is the part of leaf and how leaf actually is a pigment or is a plant that carry out photosynthesis. Because it contains chlorophyll. Yes, that's the answer. And I'm going to show you some parts of leaf and their functions. The first one that we're going to look at is the leaf stalk. The leaf stalk is like a stem. It's like a stem. And this leaf stalk, the function of this leaf stalk is to position the leaf blade in place to receive a reasonable amount of sunlight. Yeah. To position the leaf blade in place, and this leaf blade is your main leaf, that large surface area that open very wide to allow what? Sunlight. Let's look at the stomata. The stomata is responsible for gaseous exchange in plants. Gaseous exchange and the exit and the entry of water vapor. That's the function of the stomata. Then the lentice, I think the lentice is for dry woody stems. Yes, it allows gaseous exchange in dry woody stems. Then let's look at the intracellular spaces. In the intracellular spaces serve as a link for this gaseous exchange. It serves as a middleman, let's call it the middleman, for this gaseous exchange to go in and go out and the water vapors. Yeah. So there is another thing called the vascular tissue in a plant, and they are made up of your xylem and your phloem, where your xylem is responsible for water conduction across the plants. Yeah. And the phloem is responsible for food transportation across the plant. And there is another thing called the palisade mesophyte and the spongy mesophyte. The palisade mesophyte is the main site of photosynthesis in the plant. Yes, it's the main site. Yeah, and the sponges is responsible for gaseous exchange. It's the main site for gaseous exchange. Here is the reason. You know, sponges are very loose. They are very loose and they are loosely packed. So therefore, making gaseous exchange very easy. Very, very easy. And now let's talk about the chemosynthetic nutrition. So the chemosynthetic nutrition is the process whereby autotrophic bacteria manufacture their own food by the release of energy, and when that energy is gotten as a result of when they oxidize simple inorganic compound like the H2S. An example of this chemosynthetic bacteria are your sulfur bacteria, your nitrosomonas, your nitrobacter, your ion bacteria. So the chemosynthetic bacteria that oxidize ammonia to nitrites is called your nitrosomonas, and the chemosynthetic bacteria that oxidize your ammonia to nitrates is called your nitrobacter, and the chemosynthetic bacteria that oxidize your ion 2 compound to your ion 3 compound is called the ion bacteria. And that's all for the autotrophic nutrition, the photosynthesis and chemosynthesis under the autotrophic nutrition. In my next class, I'm going to talk about the heterotrophic nutrition. See you guys in my next class.